How do LFTs monitor drug-induced liver injury in clinical trials? Imagine you're testing a new medication and want to make sure it doesn't harm the liver. Liver function tests are your early warning system in this process. These tests measure specific enzymes and substances in the blood that tell us how healthy the liver is. Before starting the trial, doctors perform baseline tests to see what normal levels look like for each participant. This way, they can spot any pre-existing liver issues. Throughout the trial, LFTs are repeated at regular intervals, often every few weeks, to catch any changes early. This systematic approach helps researchers see if the drug is causing any liver damage over time. The main enzymes checked are alanine aminotransferase and aspartate aminotransferase. When liver cells are injured, these enzymes leak into the bloodstream, so higher levels suggest damage. Alkaline phosphatase and bilirubin are also measured to evaluate other types of liver problems, like blockages or impaired function. By looking at the pattern of these enzyme levels, clinicians can determine whether the injury is primarily affecting liver cells or causing bile flow issues. For example, a rise in alanine aminotransferase and aspartate aminotransferase points to hepatocellular injury, while elevated alkaline phosphatase and bilirubin suggest cholestatic injury. Researchers use specific thresholds to decide when liver injury is significant. For instance, if alanine aminotransferase or aspartate aminotransferase levels rise more than 3 times the upper limit of normal, especially if bilirubin is also elevated, it signals a potential serious problem. This is often called Hy's Law and indicates a higher risk of severe liver injury. When such thresholds are met, the trial team may pause or stop the drug for that participant and conduct further investigations. Participants with abnormal LFTs are monitored closely until their enzyme levels return to normal or baseline. Sometimes, liver injury can progress even after stopping the medication, so ongoing assessment is vital. In addition to traditional tests, scientists are exploring new biomarkers like microRNA 120 and glutamate dehydrogenase. These can detect liver injury earlier than standard enzymes, offering the possibility of faster intervention and safer trial management. Interpreting these results requires expert review from hepatologists or trained clinicians. They help distinguish true drug-induced injury from other liver conditions or temporary fluctuations. Standardized data collection and clear criteria ensure that safety assessments are consistent across different studies. Overall, liver function tests serve as a crucial tool in safeguarding participants, guiding decisions about continuing, adjusting, or stopping a drug during clinical trials. They provide a structured, sensitive, and reliable way to detect early signs of liver injury, ultimately supporting safer development of new medications. Always remember, if you're participating in a trial or concerned about liver health, consult a healthcare professional for personalized advice and assessment.